Greetings and welcome back to another rousing discussion in human biology. Today what we're going to do is we're going to talk about chapter 2. And in chapter 2, this is really kind of a chemistry discussion. I'm going to put it out there before we even start the session that I'm not going to go over in minute detail every last one of these slides because it's a really big chapter, but I'm going to kind of hit the hot spots, those parts that are really important that'll help you understand everything else. If you've taken an introductory biology course before, this chemistry is going to be very familiar to you. The depth of chemistry that we will use in human biology, it's not so deep that you'd have to understand every last bit of chapter 22 in order to be successful in this course. But I will give you some an explanation over the general aspects or properties of this chapter. I just won't be going into detail on every single part. So now that we've gotten that cleared up and out the way, let's go ahead and talk about some terms here. So matter, from what you learned in elementary school, is anything that has mass or takes up space. So anything that has some girth or weight to it and takes up space is matter. Everything is matter. Oxygen is matter. The air is matter. Nitrogen is matter. Carbon dioxide that you exhale outside of your lungs is matter. I'm matter. You're matter. So all of that stuff that takes up space and has a mass. And even though if you can't feel the mass like air, it still is considered matter. Now, all of this matter is made of what we call elements, and those are those basic chemical blocks that we find on the periodic table. For the human body, 90% of the human body is only made up of 4 different elements: carbon, nitrogen, oxygen, and hydrogen. And those 4 different elements combined in very unique ways to make the proteins, the lipids, the carbohydrates, and the nucleic acids, or the genetic material that makes up your body. So we can take your entire body, and about 90% of it, a good chunk of it, is just made of those 4 elements. So when we get down a little bit smaller, remember when we talked about the hierarchy of organization in biology, we went from the biosphere down to the atoms. The very base of that hierarchical scale is going to be the atoms. They're the smallest unit of any element that retains its physical and chemical properties. So the physical and chemical properties of that compound cannot be changed when it's on the atom level. Now, if we have an atom of carbon and we hook it up with an atom of nitrogen or an atom of oxygen, it changes the chemical and physical properties of it. So an atom itself is a molecule that's in its purest form because there's nothing else in there. So atoms actually form to make molecules. The parts of an atom, neutrons, which are neutrally charged, they're going to be in the nucleus. Protons are positively charged. They're also in the nucleus. And then the electrons, the ones that are responsible for making those bonds and forming molecules, those are going to be orbiting around the nucleus in what we call electron shells. So if we were to look at a physical atomic structure of an element, and we were to look at something like, say here, hydrogen, on the periodic table, we know that hydrogen has an atomic number of 1. So that means it has 1 proton in its nucleus. The number of electrons that hydrogen has is going to be also 1. So the number of protons and electrons and neutrons, for the most part, they match each other. Now, if this particular atom were to lose its electron, then it would carry a different charge. If it was to gain an electron, it would also carry a different charge. So not only are electrons important for making molecules, but they're also important for something being positively charged or something being negatively charged. So we have carbon, it has an atomic number of 6. We have 6 electrons out here. 6 protons in the nucleus, 6 neutrons in the nucleus. And the way that we organize these electrons is that the first shell will always only hold 2, and then the second shell can hold more than that. So the second shell can actually hold up to 6 electrons. And then we have nitrogen. So notice how first shell still only holds 2. That's the maximum amount that it can hold, but it has 1, 2, 3, 4, 5. So it's got 1 more electrons than carbon. If we go over here to oxygen, it has an atomic number of 8 protons and 8 neutrons, and we have 2 electrons in the innermost shell. Maximum it can hold is 2, and the outside we have 6 of these electrons, and that's the maximum amount that electron that can be held in the second shell. So the atomic number is always going to be represented as the number of protons. Remember, protons are found in the nucleus, and for every element, there are a different number of protons. The mass number is going to be the number of protons and neutrons. So if we go back to carbon, it had 6 protons, it has 6 neutrons, it's got an atomic mass of 12. Protons and neutrons each have a mass equal to 1 atomic mass unit. Electrons, we don't really count their mass because it's negligible, it's really small. And then electrons are always being exchanged or shared or moving from one shell to the next. So the atomic mass is, for the most part, because we say it's the number of protons and neutrons, but there are different forms of atoms. So for carbon, there are different forms of carbon where they may have 6, or maybe a form can have 7 neutrons in there. So what we do is we take all of the averages for the different isotopes of an atom, and that's what we get the atomic mass. So what the atomic mass is of carbon is really like 12.01. So it's not just a full 12, it's kind of the average of the masses of all the different forms of carbon. So these isotopes are atoms that have the same element, so carbon, but the atomic number is slightly different because of a different number of neutrons. They either have more or less neutrons than what we would normally find, and we call those isotopes. So we can take this understanding of isotopes, and we have coined this phrase radioisotopes. And these are isotopes that are really unstable because they have a whole lot or a whole lot less neutrons in the nucleus than we would like generally or normally find. And so because they're so unstable, they're going to constantly emit energy. So we call those radioisotopes. So radiation can damage cells, and it can cause cancer because it causes the internal portion of the cell, the instructions of the cell, to be lost. However, we can use radioisotopes for imaging the body, killing bacteria, sterilizing equipment, and the radiation that we use to kill cancer cells is also done through radioisotopes. So here we're looking at some medical uses for low levels of radiation on here, for imaging that we can see using these isotopes. We're doing a brain scan, and then we have a thyroid. So, and I apologize for what's happened on this slide. I'm not really sure why this did this. Big things I want you to take from this is that molecules are atoms that are bonded together, and that's what that is supposed to say, atoms. Molecules bonded together. Compounds are molecules that are made of different atoms. So you have different atoms like hydrogen and oxygen bind together to make this compound. Molecules would be like O2, it's the same atom, or sometimes it could be different atoms. Two types of bonds that can form between atoms are called ionic bonds or covalent bonds. And we'll talk a little bit about ionic bonds here in a second. So ionic bonds are most stable when their outermost shell of electrons is full. And depending on what number shell you have, that determines how full that outermost shell can be. When we have an ionic reaction, what happens is that atoms either donate or take electrons in order to fill that outermost shell. Every atom, their goal in life is to be made whole. They want to have their outermost shell completely filled. So depending on whether or not you gave an electron away or you took an electron, that's going to determine what your charge is. So if you are a hydrogen atom and you gave your electron away so that you don't have anything in your outermost shell, then you carry a positive charge. If you are an oxygen atom and you take an electron, well, oxygen don't have to take electrons, but if you are a carbon atom and you take electrons, then you would carry a negative charge because you've taken electrons. So look at it this way. You give something away, you feel good about yourself, so you carry a positive charge. So if you give an electron away, you feel good, you give it a positive charge. If you take an electron, you feel bad about yourself, so you carry a negative charge. And really, the scientific basis behind that is that electrons are negatively charged. So if you acquire more of these negative things, then your overall charge will become negative. If you get rid of these negative chains, then your charge becomes more positive. So here is a great example of ionic bonding that's happening in some table salt that we use quite frequently. You have your sodium ion over here. it only has one electron in its outermost shell. It needs to acquire several more in order for the outermost shell to be full. So it's just easier to give this one electron away to something like a chloride ion. So that chloride ion takes that electron. It's going to carry a negative charge because it took something. It feels bad about itself. And then the sodium ion, because it gave something away, now only has 2 valence shells, and its outermost shell is now full because it got rid of that one electron. It feels good about itself, so it carries a positive charge. Or remember, the more scientific way to explain that is that it got rid of something that was negative, so it has less negativity, fewer electrons, so the charge elevates. And the bond that's formed between sodium chloride we call an ionic bond. So remember, in ionic bonds you have a transfer of electrons. Something is being taken, or something's being given away or donated, and something's being taken. Covalent bonds, on the other hand, this is another way that electrons or atoms try to fill their valence shell, but instead of giving anything away or taking anything, they're just going to share it. So the great example of a covalent bond would be that in water, H2O. So each electron or each atom is going to contribute at least one electron to that shared pair, and they just spend equal time traveling around. So water is a much more stable molecule than hydrogen is on its own, because hydrogen only has 1 electron. It requires at least 2 electrons in order for its outermost shell to be full. It's much, much easier to just share its 1 electron with oxygen in order to fulfill its outermost shell being full. So they could be, remember I said before, you need to share at least 1 electron. Sometimes there are covalent bonds where 2 pairs of electrons are shared, and even triple covalent bonds where there are 3 pairs of electrons that are shared. And the way that they kind of would depict that is either 1 line or 3 lines, depending if it's single, double, or triple. So here we have that oxygen molecule over here, and we can fit 8 electrons in this outermost shell. We can fit some more electrons in this outermost shell. So this hydrogen, it wants to bind with oxygen. It's just more favorable to make this stable water molecule, because now everybody's outermost shell is completely full, even if it's just for part of the time. With 2 oxygen molecules, they can form a double bond, and the reason we have a double bond is because there's 1, 2, that's 1 pair, 3, 4, that's another pair. There are 2 pairs of electrons that are being shared there. So water, because of its covalent bonding and because the way that water shares its electrons is a little uneven with hydrogen, it's considered to be a polar molecule. So that means that it has a partially positive part, and that's going to be where the hydrogens are, because it spends a little less time with the electrons. So it's kind of giving that electron away a little bit more than the oxygen is. And then you have the oxygen that's got a partially negative charge, because it kind of spends more time with that electron than the hydrogen does. It's kind of like if you had twins, right? And they're supposed to share a car, and then you have one twin that spends more time with the car than the other twin does. And so hydrogen is that twin that says, you can spend a little bit more time with the car. I'm okay with that. So it carries a partially positive charge. The oxygen is the twin that says, I want the car all the time. I'm going to spend more time with it. He kind of feels a little bad about himself because, you know, he takes the car from his twin sister or twin brother or whatever more time, so it got a negative charge. It has a negative charge. Water molecules are what we say to be polar. Now, because water molecules are polar, there's some really cool things that can happen with that. Water has bonds that can form with other water molecules that we call hydrogen bonds. Those hydrogen bonds allow for really cool things to happen. That water is a molecule that can take a lot of heat or temperature and a lot of heat and just have a really small increase in temperature on there. Water can also, it has a cohesive aspect to it because of these hydrogen bonds. So because of those hydrogen bonds, there's going to be cohesion and forces between there. So because of the polar nature of this water molecule, and when we say polar, remember that's what this little symbol stands for, is partial. We have a partial negative in at the oxygen and a partial positive in at the hydrogens. So we form the attraction forces called hydrogen bonds between this positive N and this partial negative N and this partial positive N and this partial negative N. So we have these hydrogen bonds that are formed. Because of those hydrogen bonds, just as a repeat here, really cool things that we can have. Water is a liquid at room temperature. It has a high heat capacity, which means it can take a large amount of heat and just have a very small change in increase in temperature that makes sure that your body temperature stays relatively stable because we're mostly made of water. It has a really high heat of vaporization, and that allows for evaporative cooling, and that's why sweating kind of cools us off. So other really cool aspects of water is that it is a solvent. It is a solvent because, can you guess? Can you guess? Yes, the polar nature of water molecules. So because it has this partial negative N and this partial positive N, anything that has a charge to it, like, oh, say this sodium ion, which remember on the previous slide, gave away its electron, so it feels good about itself, carries a positive charge, we can have these partial negative parts of the oxygen kind of corral around it. So when you put salt in water, what's physically happening is the ionic bond between sodium and chloride is being broken apart by the water. It's dissociating, and it helps to facilitate chemical reactions when we can have that dissociation take place. So because water is a polar molecule, we have a polar molecules attract water, so they're hydrophilic. Hydrophobic molecules do not attract water, and they're nonpolar. So in nonpolar covalent bonds, the electrons are shared equally, and there are no charges that are there. So water, remember, that's a polar covalent bond. And because it is a polar covalent bond, it makes it a great, or what we call a universal solvent on there. Vegetable oil is nonpolar. There's no partial positive or partial negative parts, and since there are no charges associated with it, it doesn't really dissociate. And that's why oil and water do not mix, because vegetable oil is nonpolar. There's no charge to it. There's no partial positive part, no partial negative part. So that partial positive hydrogen and that partial positive or negative oxygen, they have nothing to be attracted to. So they just kind of separate from one another. So that's why we consider oil to be hydrophobic, or it's just repels water. So we have this cohesive property of water, and that is because of the polar nature of water. Water molecules cling to each other through these hydrogen bonds. Adhesion are water molecules clinging to surface, and they will cling to the surface like the inside of your blood vessels, which is a good thing that we want to do. It allows for less turbidity, more fluid flow through the blood vessels, which we like to have, and it helps to maintain pressure. Frozen water, which you know already, an ice cube is less dense than liquid water, so as a result, that ice is going to float, and this fact that ice floats can act as a bit of an insulator. Next thing we're going to talk about are acids and bases, and once again, I apologize, I don't know exactly why this looks this way. Acids and bases are going to be the dissociation of hydrogens, or actually the accumulation of hydrogen ions. So where you see lots of hydrogen ions, it's going to have a lower pH, and we consider that an acid. Your pH scale is going to run from 1 to 14, with 7 being neutral, and the standard bearer for neutral is water. Anything less than 7 is an acid, and anything higher than 7 for the concentration of hydrogen ions is lower when you're higher than 7. Anything higher than 7 is a base. So we can have different types of, so hydrogen chloride, go back to this slide really quick. Hydrogen chloride is a very strong acid, and we have sodium hydroxide is a very strong base. This is all stuff I've kind of already said before. So we're just going to move on through that. So the pH scale, 0, usually we use 1. We very rarely see things, at least in human biology, that have a pH of 0. But 1 to 14, water being the neutral. Anything less than that is going to be considered an acid. Anything higher than that is considered a base. Now, the pH of your blood is 7.4, so the pH of your blood is slightly basic. And here are just some few common household items or common things that we find for what their pH is. So a buffer is a solution that resists changes in pHs when acids or bases are added into it. Now, buffers are really important in your body because they help to make sure that the pH stays within a narrow range. So we have something called carbonic acid, and it helps to act as a buffer against the blood in much the same way that bicarbonate ions do. So bicarbonate ions can either, bicarbonate and carbonic acid can add hydrogen ions in order to bring the pH down somewhat if it's too high, and then bicarbonate ions can pick up hydrogen ions in order to increase the pH on there. So two atoms that you're going to see a lot are carbonic acid, which is what this is supposed to say if it was not doubled up, and bicarbonate ions. Those are two ions that are buffers in the human body that will either take up or donate hydrogen ions to make sure that the pH of the blood stays within that range of about 7.4. So now that we've talked about some very basic fundamental chemistry, we're going to kind of go into what we consider biochemistry. So these are going to be a discussion of the four classes of macromolecules or organic molecules that we find in the body. Now, when we say organic in biology, what we mean by that is that these are molecules that have a carbon backbone. So they're in some sort of form of that structure, somewhere there, we're going to have a carbon atom. So when we say organic in biology, we're not talking about, not grown with harmful pesticides or anything like that, which is really awesome. And I encourage you to buy or grow organic as much as you can. But when we talk organic, scientifically speaking in biology, we're just saying that there is carbon associated with it. So there are four different types of macromolecules that are organic molecules that are made up in your body: carbohydrates, lipids, proteins, and nucleic acids. So to make these macromolecules, they have to be built from something smaller, right? And that something smaller that they're made up are what are called monomers. Carbon has its own specific monomers that make up carbohydrates. Proteins have their own specific monomers, and they're called amino acids that make up proteins. Lipids have their own specific monomers that make up lipids or fats, and nucleic acids are the same way. They have their own specific monomers that make up nucleic acids on here. And the way that those things are made up is what we call through a synthesis process. The synthesis process that makes up all these macromolecules is called a dehydration synthesis, means it loses water. And then to break it down, we add water, and that's called a hydrolysis reaction. So to break it down, dehydration, so to take a carbohydrate and break it down into glucose, those smaller subunits, we remove water. To take glucose and put it into a carbohydrate, then we have to add water. So carbohydrates is a major energy source for our body. In plants, it can be structural. So cellulose helps to make the cell walls of the plant and allows for it to have structural aspect to it. Carbohydrates are made of carbon, hydrogen, and oxygen. And the building block of carbon or carbohydrates is some sort of sugar, or what we call monosaccharides. So monosaccharides will bind together via dehydration synthesis. We're going to move the equivalent of a water molecule, two hydrogens and an oxygen. And then we have, that's our dehydration reaction that takes this single sugar and has now made it a dual sugar. So that's called a disaccharide. When we have lots and lots and lots of these sugars piled together, then we have a polysaccharide. So complex carbohydrates or polysaccharides, there are lots and lots of sugar molecules that are linked together. And we say that it is a long polymer of glucose molecules. Now, because we have all these sugar molecules together, and one of the cool jobs of sugar is that it provides energy for your cells, is that the longer the sugar molecule is, the longer the energy storage we have. So starch is the energy storage in plants, and in our bodies, we take our polysaccharides and we store them in our liver and store them in our muscle cells, and it's called glycogen. Cellulose is that structural polysaccharide that we find in the plant walls. We don't have an enzyme to break down cellulose, so it's just like fiber for us. It's just roughage. It helps to kind of clear out the digestive system. But there are some animals, like cows, that do have the enzymes to break down cellulose. And you notice cows like to chew grass all day long because they're getting a lot of really good energy out of it. It's a really good polysaccharide for them. So the next macromolecule we're going to talk about are lipids. And we've already said this, that oil and water do not mix. And the reason that oil and water do not mix is because water is a polar molecule and lipids are nonpolar. There's no partial positive end of a lipid, so there's nothing for them to be attracted to. Now, lipids are very important in your body. They have a very diverse function of it for themselves. So you have steroids that are technically considered lipids. So you think of testosterone, estrogen, and even cholesterol. Those are steroids. Phospholipids, which make up the plasma membrane or that boundary on the outside of your cell, and triglycerides, which our body uses to store fats. There's been a lot of discussion here recently on how our bodies and our brains really need more good fats than we thought we did before. There was this whole push to kind of get rid of fats in our diet, and we see more and more how important healthy types of fats are. And healthy types of fats would be kind of unsaturated fats and not the trans fats. Trans fats aren't very good at all because they're basically man-made, and your body doesn't really know how to handle breaking them down. So triglycerides, or we would normally consider dietary fats, have one glycerol molecule, and hence the term tri, 3 fatty acid tails. Great energy storage for your body, provides insulation for your body. It's called adipose tissue, which we'll talk about throughout this course, provides cushioning, and these two forms of fats come in two different, these triglycerides come in two different forms. They can be fats or they could be oils, or what we would typically say saturated fats and non-saturated fats. Saturated fats or fats usually come from an animal. So that would be things like butter and lard. And then oils, they originate from a plant. So things like olive oil, sesame seed oil, grapeseed oil, those are going to be more plant origin. Fats are solid at room temperature. Oils are liquids at room temperature. Oils are what we consider non-saturated fats. Fats are saturated fats. So here's a picture of that triglyceride. We have our glycerol head and we have our 3 fatty acid tails. And to hook these two together, we still have dehydration synthesis, moving the equivalent of 2 hydrogens and an oxygen. To break it down, hydrolysis, we have to add water molecules in order to break apart that glycerol and fatty acid tail. So triglycerides, which you already knew, were going to be hydrophobic because oil and waters do not mix. They would have a tendency to clump together in body fluids. Because they are nonpolar, what happens is that in order to break them down, you need emulsifiers like bile to help surround the triglycerides and sort of disperse them so that they can be broken down. Waxes are one fatty acid attached to an alcohol, and waxes prevent the loss of moisture from body surfaces. So your earwax that is released from your cerebrum glands in your ears, they actually form this wax here. So it's really just a form of lipid, and it helps to kind of attract things that shouldn't go any further into your ear canal. So fatty acids are long chains of carbons and hydrogens. They're always going to end in our carboxyl group on there, COOH, and they contain 16 to 18 carbons. Those fatty acid tails can be saturated, like those fats that come from animals, or they can be unsaturated, like those oils that come from plants. Saturated fats have no double covalent bonds between the carbons, so they have the full complement of hydrogens. That's why they are called saturated. They are saturated with as many hydrogen molecules or many hydrogen atoms they can possibly have. Unsaturated fats have one or more double bonds, so there are some hydrogens that are missing. So because we have these double bonds, it allows it to be much more fluid because there are kinks in the chain. So there are bends because of those double bonds, and as a result, it's going to be more fluid at room temperature. So that's why oils are considered unsaturated fatty acids, and animal fats, because they are saturated with carbons, are saturated fatty acids. So saturated fatty acids, diets in high accumulation of saturated fatty acids for individuals that have sedentary lifestyles, or for individuals that have there's nothing they consume except for saturated fats, has been known to lead to arteriosclerosis, which is a disease of blood vessels. Now, that's not to say that saturated fats are always all bad. If you have an active lifestyle, so that you are going for a walk at least 30 minutes a day, if you're even playing with your little kids, you're working out at the gym, saturated fats are actually really good for your body, and it's an excellent way for your brain to get the energy it needs and your body to get the energy that it needs without having to depend so much on carbohydrates. And we are, especially in Western cultures, we are very carbohydrate, have very high carbohydrate diets. But that is not to say if you are just a couch potato and you don't really get up to do much of anything except for find the remote control, then you probably don't want to have a diet that's really high in saturated fats. You might want to use more unsaturated fats, but in all things, moderation. Trans fats are completely man-made, and these we know are highly correlated with heart disease more than any other types of fat. So what trans fats are is that they take an unsaturated fat, like a corn oil, right? And corn oils aren't always like the greatest oils for you to have because they have to do so much work and so much processing to get the oil out of it. Whereas olive oil, it's very easily to just press it out that you can get that oil out easier, that oil is more refined, it's more pure, that's a better oil for your body to have as opposed to like corn oil or any like some of those seed oils. It's much more difficult to get it out of there. So with the trans fat, they take an oil like a corn oil and they're unsaturated, but they've done so many things to it. They're missing hydrogens and opposite ends of those carbon chains that they've really like changed the chemical structure of the fat, and your body no longer recognizes it as something that it can metabolize very easily. So it gets stuck into your blood vessels much easier even than saturated fats. So in the past, the recommendations for the total amount of fat in a 2,000 calorie diet was 65 grams. Now, through research indicates that the type of fat that is eaten is more important than the quantity of it. So if you have fats, if you're going to consume fats, which I'm a person that I love fats, I like olive oil. I use olive oil for pretty much every meal that I cook. Having ghee every now and then, which is an animal fat, is a good type of fat for you to have. And even butter, particularly grass-fed butter, is an excellent fat for your diet because you are getting lots of energy from it, and then you're also getting really good molecules and atoms from these types of saturated fats. And olive oil is my personal favorite, but grapeseed oil is a really good oil and fat to have as well. So fats are not terrible. Your body absolutely needs fats. And the more fat that you consume, the good types of fats, like the oils that we talked about and the butters and ghee, those really, remember talking about grass-fed butter there, that it actually will keep your body from having to rely so heavily on carbohydrates, which actually turn into adipose tissue that we never actually use sometimes if you have a very sedentary lifestyle. So understanding a food label. So we're going to take all the stuff that we just talked about, that we learned about chemistry, atoms, and molecules, and these different types of macromolecules with carbs, lipids, nucleic acids, and proteins, and see how we can apply this information to a food label. So the very first thing that you're always going to see in the food label is that I always read the serving size, because just because it's a little bag of chips, it might have what they would consider more than one serving size in there. So for this particular label, so you want to make sure that is the serving size one bag, or is it just half a bag, or is it just 13 chips, or is it just 13, a cup of this or whatever. So read the serving size there, because all of these calories and everything that's on here is going to be based on the serving size. So that's a very important portion. So for this particular food, it has 2 servings per container. So if you eat the entire bag, even if it's something good like trail mix, if you eat the entire bag, you're going to have to double these numbers. So instead of getting 250 calories, you're actually getting 500 calories if you eat the whole bag. That's why it is highly recommended that if you're going to have a snack, that you just pour some out into a bowl or a separate container, because we have a tendency to eat until we reach the bottom of the bag. And that's when those calories double, and that's when we're really overeating, and a lot of times we don't realize it. So let's say we stuck to this serving size. We have 1 cup. There are 250 calories in that cup. The number of calories from fat is 110 calories. What type of fats we have in here? We have saturated fats, no trans fats on there. We have some cholesterol. There's some sodium in there. these types of items, the fats, you do want to have them limited, especially the sodium on there. You don't want to have too much sodium. In fact, this tells you that this product alone is going to give you 20% of the amount of sodium that you need for the day. So you want to sort of be mindful of that. Down here we have carbohydrates. Fiber is a really good carbohydrate because it acts as roughage on there, but we don't get any in this product. We do have some sugars, so there's 5 grams of sugars on here, and those sugars, every time you see carbohydrate, your brain should automatically say sugars because carbohydrates are just a long chain of sugars that are on there. And then it even has different vitamins that will be found in there, and then you have a little bit of proteins in here. So there are different types of diets that we have, like the Atkins diet was heavy protein, low carbs. The diet that's kind of like Atkins on steroids is the not paleo diet, but the ketogenic diet, and that's very high proteins, very high fats, limited carbohydrates. So you would want to look for how much proteins are in there. So other types of fats that your body needs are phospholipids. They are very structurally similar to a triglyceride, but instead of having 2 fatty acid or 3 fatty acid tails, it only has 2. And that third fatty acid tail is replaced by a phosphate group. So when we look at a phospholipid, it's got a hydrophilic part to it, so the head likes water, and then it's got hydrophobic tail. The tails don't like water. Now, why phospholipids are so important is because they make the plasma membrane of each and every one of your cells. And because there's this hydrophilic portion to the plasma membrane and this hydrophobic portion to the plasma membrane, each one of your plasma membranes is going to be dual layered, or what we considered a bilayer. And the way that bilayer is situated is that these hydrophilic heads are always going to be near aqueous solution or near water, and the tails are always going to be tucked nicely in the center because they don't like the water. They're hydrophobic. The tail has that PO4 minus, which has a charge to it. Since it has a charge in that phosphate group, it's going to be hydrophilic and like to be associated with the water. Other type of fats, as we said before, steroids. Typically 4 fused rings with a functional group that are attached to them. Cholesterol is the base steroid from which other steroids such as estrogen and testosterone are made from. And you also even have cholesterol in your plasma membrane. On to on to proteins. So proteins have a lot of really cool functions. They can be supportive. That would be things like the keratin that makes your hair and nails, and the collagen proteins that form underneath your skin and help to support your ligaments. They can have enzymatic properties, which means that they increase the rate of a reaction. So an example of that would be like salivary amylase, which is an enzyme that's in your mouth, and it helps to break down carbohydrates. FYI, anything that ends in ASE when we're talking biology is a enzyme. So in amylase, proteases, all of those are enzymes. Every enzyme that you ever come into contact with is a protein. But not all proteins are enzymes. All enzymes are proteins, but not all proteins are enzymes. We can have proteins that transport things across the plasma membrane. So the sodium potassium pump is a protein transporter that helps to exchange sodium for potassium ions. Super important in neural function as well. Proteins can be defensive. So we have antibodies that are proteins that bind to foreign substances, and these foreign substances are called antigens and will help disable them. So we need that in the lymphatic system, and also in your blood you have these antibodies. They can be chemical messengers. They can be contractile proteins. So the cilia that's in your respiratory track, they move and have a motion of what's called the ciliary escalator, and those are made of proteins as well. So what makes up proteins are what we call amino acids. They're the building blocks of proteins. So the building blocks of carbohydrates, remember, were sugar molecules, things like glucose. The building blocks of lipids were fatty acid tails and a glycerol molecule. The building blocks of proteins are amino acids. So when we have amino acids bond together and make this polar covalent bond, which means there's going to be a partial positive side and a partial negative side, that's called a peptide bond. To make a polypeptide, there are several amino acids that are going to be linked together. So proteins have a really interesting, because there's so much things they have to do. They have a three-dimensional shape that mimics what its function should be. So protein form dictates function. How a protein is shaped determines how it functions. If something like the pH or the temperature is not quite right, it will cause a protein to lose its shape. And if it loses its shape, it loses its function. The process of a protein losing its form and thus losing its function is what we call denaturation. And that just means that the protein's ability to do what it needs to do is now been disrupted because it has unfolded from its three-dimensional shape. So there are four different levels of protein structure. We have the primary structure, which is just the linear sequence of amino acids and how they link together and form these peptide bonds between amino acids. And then the secondary structure is where it either folds into these sheets that we call beta sheets, or it can form these little curlicues that we call alpha helices. The tertiary structure is when all of those beta sheets and those alpha helices form and bond together. And then what kind of helps to stabilize it are these sulfur to sulfur bonds that we call disulfide bonds. If we take all of these tertiary structures, or at least two of those tertiary structures, and we form them together, then we have a quaternary structure. Good transporter protein, important protein, is that has this really cool quaternary structure is hemoglobin. Hemoglobin is found on the inside of your red blood cells, and its job is to carry oxygen in the red blood cells and carry a little bit of, it's called carbanohemoglobin, a little bit of carbon dioxide back to the lungs. So hemoglobin has a beautiful quaternary structure that is kind of like the standard bearer, or the supermodel of quaternary structures, the way that it looks. And it has a nice little iron molecule in the middle there that has this affinity or binding ability for oxygen. All right, so our very last macromolecule that we're going to talk about are nucleic acids. Nucleic acids are polymers, which means they're just large molecules that are made up of small molecules, and those smaller molecules are called monomers of nucleotides. A nucleotide consists of a phosphate group, a 5-carbon sugar, and a nitrogenous base on there. So the functions of these nucleic acids are to store information, such we see as in DNA. And RNA contains the instructions for how things are supposed to happen within the cell, and it also helps to conduct chemical reactions that we see for ATP. So here we have that 5-carbon sugar, 1, 2, 3, 4, 5 carbon sugar, and we have a phosphate group that's attached to it, and we have this nitrogenous base that contains some sort of carbon. So this is just a very generic nucleotide. So when we have these generic nucleotides bind together, then we form nucleic acids. So probably the most popular or the sexiest of the nucleic acids on here are going to be DNA and RNA. Some differences that we see between the two of them: they both have the 5-carbon sugar, the nitrogenous base, and they have the phosphate group attached to it. But the sugar in DNA is called deoxyribose, in RNA it's called ribose. The bases, those nitrogenous bases, which over here we just said, it's a generic base. We haven't said exactly what that base is. But the nitrogenous bases that we find in DNA are adenine, guanine, thymine, and cytosine. For RNA, it's slightly different. We don't have a thymine. We replace that with uracil. How it's kind of formed, or what it looks like, what its three-dimensional form is that's a double-stranded helix with a base pairing. For RNA, it's just single-stranded. And if it forms a helix, yes, it does in DNA. Nope, it doesn't do that in RNA. So RNA is really meant to be messenger between the DNA and how the proteins are made. The DNA instructs exactly what proteins should be made, and it's the RNA that takes that message out to ensure that those proteins are made according to the instructions of the DNA. So here we have that double-stranded helix, and we have that complementary base pairing where adenine always pairs with thymine in DNA, and cytosine always pairs with guanine in DNA. And then we have this sugar-phosphate backbone, phosphate, sugar, and then the nitrogenous bases make up the inner portion of the DNA. And if you thought about unwinding it and imagine it as a ladder, then these nitrogenous bases, where A pairs with T and C pairs with G, would be like the rungs on the ladder. So we've talked about kind of the rock stars of nucleic acids, but a character doesn't really get a whole lot of press as a nitrogenous base-carrying nucleic acid is ATP. ATP is super important, and we're going to talk about ATP for the remainder of the session because it is your energy carrier. The reason that I am able to give you this high energy lecture is because of my ATP. The reason you're able to contract your muscles is because of ATP. The reason that you're able to make any enzymes or to have dehydration synthesis or hydrolysis or any of the stuff that your body needs to do to stay alive is because of ATP. So with ATP, what we're looking at is it's adenosine. That's a nitrogenous base over here. Remember we talked adenine, so it's a adenosine. It's just a form of adenine. That's the nitrogenous base. We have adenosine and we have 3 phosphate groups. Now, phosphate is PO4 minus. It's negatively charged. So if you take 2 negatively charged entities and pop them together, that's going to be a very strenuous relationship because like charges tend to repel from one another. So with ATP, not only do we have 2 negatively charged phosphates hooked up together, we got 3 negatively charged phosphates hooked up together. And so there's a lot of energy that we have there. When we break the bond between that second and that third phosphate, there is a tremendous amount of energy that's released. And that tremendous amount of energy that's released is what we use to power muscle contractions, make micromolecules, have nerve conduction take place, power the sodium-potassium pump, which is vitally important in your nervous system. So now we have this explosion of energy that's taking place, and now we're not left with ATP, we're left with ADP. Now look at how sweet this science is. ADP gets recycled. We don't just throw it away. All we need to do is pop another phosphate group on there. So we can add another phosphate or phosphorylate ADP into ATP. And the way that your body does that is that when you eat and use oxygen, you go through cellular respiration. Cellular respiration is a 3-step process where it turns ADP, ultimately turns ADP into ATP. And there are different types of phosphorylation that cause that to take place. And this cycle just continues. Pretty awesome biochemistry that happens there. We are literally barely just scratching the surface of what goes on with ATP and why it is so freaking important. So here is that adenosine on there. We have our 5-carbon sugar, and then we have our 3 phosphate groups that are attached to it. When we have that bond between that third and the second and third phosphate break off, we have a tremendous amount of energy that's released, and we're left with ADP that gets recycled through cellular respiration and sometimes even fermentation. We take that ADP, phosphorylate it, turn it into more ATP to power those muscle cells, to power those chemical reactions in your body, and everything you need to do to stay alive and stay beautiful. Woo! So that is all of chapter 2. The next time that we speak to one another, unless you're going to listen to this as a recording, we will be going over chapter 3 and then 4, where we're going to get a chance to actually get into some human organ systems and talk about tissues. I hope you enjoyed this lecture as much as I've enjoyed giving it to you. Have a fancy day. See you soon.